Welcome back to Microbiology, Chapter 9, Part 2. In this video, we'll look at biofilms, then move into oxygen requirements, and finish with the effects of pH on microbial growth. Most microbes in nature don't actually live as isolated cells. These are also known as planktonic cells. Instead, they usually live together in biofilms, and these are structured communities of microbes. And they are everywhere. Biofilms are found in pond slime, slippery rocks, dental plaque, catheters, lung infections and cystic fibrosis, and even in endocarditis. They also even form on the International Space Station. They can clog hoses and equipment. Because of their protection, biofilm-associated microbes are harder to kill and more resistant to antibiotics. So new strategies focus on preventing microbial adhesion to surfaces rather than trying to kill cells that are already present in biofilms. These biofilms or communities are encased in EPS, and that stands for extracellular polymeric substances. And these make up 50 to 90% of the biofilm's dry mass. EPS is mostly polysaccharides, but can also include proteins, nucleic acids, and lipids. And this structure provides major advantages, such as physical protection from drying out, from, for example, human immune defenses, and protection from antibiotics. We also have nutrient channels in biofilms, and this allows them to transport food and waste, as well as shared metabolism, where one species' byproducts might feed another species within the biofilm. As I mentioned earlier, biofilms cause persistent problems in healthcare, such as dental plaque, catheter infections, antibiotic resistance. Microbes in biofilms are much harder to kill because the EPS blocks antibiotics, and because some cells are going to be in a slow metabolic state. So new research, as I mentioned, focuses on preventing microbial adhesion to surfaces rather than trying to destroy established biofilms. Quorum sensing is cell-to-cell communication using molecules known as autoinducers. This lets microbes sense their population density and coordinate virulence or dispersal. So I'll talk more about that soon. And finally, we're going to look at biofilm formation in the next slide. So we start with planktonic cells. Remember, these were free-floating cells in an aquatic environment. They're going to become attached. And when they're attached, they're called sessile cells. And we'll start growing an extensive matrix with water channels and the formation of the EPS. So that was extracellular polymeric substances. Finally, some cells at the periphery will revert to planktonic form, and they're going to leave the biofilm to colonize new sites. Quorum sensing occurs within biofilms, and this is cell-to-cell communication between the microbes within biofilm using autoinducers. And autoinducers are different depending on if the bacteria are gram-positive or gram-negative. So in gram-positive bacteria, they tend to use short peptides, whereas in gram-negative, they use N-acetylated homoserine lactones. And I'm not going to test you on this, so you don't have to know these or the structures. Just know that gram-positive and gram-negative bacteria use different types of autoinducers in quorum sensing or cell-to-cell communication. All right, let's recap 9.1, and this will also include material from the first video. So the first is generation time. That was also known as the doubling time, and it's the time it takes for a bacterial population to double in number through one round of binary fission. And the second one, identify and describe activities of microorganisms undergoing typical phases. So the growth curve, we're looking at the growth curve. Lag phase is when the cells are metabolically active, they're repairing damage, making proteins, but they're not really dividing yet. Then we have that log phase, that spike in growth. That's the exponential phase. There's rapid binary fission. There's an exponential increase in cell number, uniform metabolic activity, and this is also the time where they're the most sensitive to antibiotics. Then we had that flat phase, right? The stationary phase, where nutrients are depleted, waste has accumulated, and growth rate equals death rate. This is also when endospores, virulence factors, or other metabolites might form. Finally, the fourth one is the decline or death phase, where cell death exceeds growth, but some persisters might survive. For For the third one, so examples of methods used to determine viable and total cell counts. So we talked about direct microscopic cell counts, like through that Petroff-Hauser chamber with the grids, and you count each of those square grids. We talked about viable plate counts using pore plate or spread plate or membrane filtration, where we count colonies and calculate colony forming units per milliliter. We also talked about the most probable number method for dilute samples like water testing, as well as some indirect methods such as measuring turbidity or cloudiness of a sample through the spectrophotometer. We could also do dry weight as a part of indirect methods, or metabolic activity. For the fourth bullet point, examples of cell division not involving binary fission. So budding was a small outgrowth or bud that forms from the parent cell and detaches later, such as we see in yeast. Fragmentation is when filaments split into multiple cells that separate and grow independently, such as cyanobacteria or filamentous bacteria like the actinomycetes. Let's see, the next one, biofilms. So we just talked about biofilms. They start with planktonic or free-floating cells that eventually attach to a surface and become sessile cells. These produce that EPS, the extracellular polymeric substance, and start forming these clusters of microbes. They form water channels and nutrient channels, and the biofilms really protect them from dehydration, from the host defense or immune system, and antibiotics. For the sixth one, health risks. So health risks include like dental plaque, chronic wounds, cystic fibrosis, lung infections, or device-related infections like catheters, artificial joints, even contact lenses. So bacteria and biofilms, we saw that they have increased antibiotic resistance. So prevention or control can include device replacement, or surface modifications to prevent attachment, or specific targeted antimicrobial drugs that can penetrate biofilms. Finally, quorum sensing. So quorum sensing is when microbes release and detect signaling molecules called autoinducers. And when they reach a certain threshold density, autoinducers can trigger changes in their gene expression, so they make different types of proteins or enzymes. So this could regulate virulence factors, biofilm development, or their dispersal if they leave the biofilm, as well as antibiotic resistance. All right, now we're going to switch gears and move into oxygen requirements, including classification of microbes by their oxygen tolerance. So we tend to think that oxygen is everywhere, and it generally is, as humans and other animals require oxygen, and oxygen makes up about 21% of the Earth's atmosphere by volume. But there are areas that are pretty much devoid of oxygen. So our book gives us some examples, such as bogs. This is a type of wetland that's really has acidic soil, and the undisturbed sediment will be pretty much anaerobic. They also give us the example of a cow's stomach, which is devoid of oxygen or oxygen-free. These are areas where obligate anaerobes can survive. These are bacteria that cannot live in the presence of oxygen. Oxygen is toxic to them. Think of in our bodies as well. In human bodies, anywhere where there is no blood flow will tend to be anaerobic. So maybe think about the stomach, or other regions where there are no blood vessels, no hemoglobin to deliver oxygen. Early Earth lacked oxygen, so the oldest bacterial lineages were anaerobic. Oxygen only became common after photosynthetic cyanobacteria began producing it. And oxygen is very useful, but also can be dangerous. It can form reactive oxygen species, or ROS, such as superoxide, peroxides, hydroxyl radicals, and these damage our proteins, our lipids, our DNA, RNA, and other cellular processes such as inactivating coenzymes or disrupting our electron transport chains. So in order to survive in the presence of oxygen, cells have to be able to produce enzymes to detoxify these ROS, these reactive oxygen species. And these enzymes are enzymes such as superoxide dismutase, catalase, and or peroxidase. Obligate anaerobes cannot survive in the presence of oxygen because they usually lack these enzymes and they're killed by oxygen. So here's how we classify microbes by aerotolerance or oxygen tolerance, and much of this overlaps with what we've done in lab already. Obligate aerobes require oxygen because they need it for aerobic respiration. Examples include Mycobacterium tuberculosis and our skin bacterium Micrococcus luteus. These will only grow at the top of a thioglycolate tube. Obligate anaerobes are the opposite. They are killed by oxygen, right? They lack those enzymes to get rid of those reactive oxygen species. They will only grow at the bottom of a thioglycolate tube. So they perform anaerobic respiration or fermentation, no aerobic respiration. And examples include those endospore-producing species, such as Clostridium or Clostridioides. Facultative anaerobes grow with or without oxygen. They actually prefer oxygen, so they actually make more ATP under aerobic respiration, but they can switch to anaerobic respiration or fermentation when there is no oxygen available. So examples include Staphylococcus or the Enterobacteriaceae family that live in our gut. They grow throughout a thioglycolate tube, but they're going to grow denser at the top of the medium. Continuing on, we have our aerotolerant anaerobes. These don't use oxygen but tolerate it, and they rely on fermentation only. So examples include lactobacilli or streptococci, and they're going to show even, sparse growth throughout a tube, like the semisolid agar deep or the thioglycolate. These are not the same as obligate anaerobes. Remember, these are killed by oxygen because they don't have those enzymes to neutralize or get rid of reactive oxygen species. Whereas these can tolerate oxygen. These must have at least one of those enzymes to get rid of those reactive oxygen species. Microaerophiles need low oxygen concentrations. Remember that the atmosphere was about 21% oxygen, so they need less than that. Examples include Campylobacter jejuni, which causes gastrointestinal infections, and they're going to show a thin band just below the surface of the tube. These, again, undergo aerobic respiration, but best at low oxygen concentrations. Finally, we have capnophiles. Capnophiles prefer high carbon dioxide concentration, often in combination with low oxygen. Examples include Neisseria species, and these are aerobic bacteria that are capnophilic. So usually you'll see this term, aerobic capnophilic bacteria. So they do aerobic respiration, but they prefer having high CO2 environments. Thioglycolate medium or broth is a differential enrichment medium that's used to study and grow bacteria with different oxygen requirements. In this medium, the top of the tube will have the highest oxygen concentration, pretty close or equal to atmospheric levels, and the bottom of the tubes will be anaerobic, so there will be no oxygen or very insignificant levels. We'll usually use some kind of oxygen indicator in there, and we'll see this in lab 17 called resizurin. That's pink when there's oxygen present, so we know where in the tube oxygen is present. And we're going to see that obligate aerobes grow at the top of these tubes. Obligate anaerobes grow at the bottom. Facultative grow throughout, but densest at the top. Aerotolerant grows throughout, but sparse everywhere. And microaerophiles will grow just below the top, where oxygen concentrations are lower. So thioglycolate medium is a great lab tool for determining bacterial aerotolerance types. Here's another more realistic depiction of thioglycolate results from the American Society for Microbiology. This often happens, this note at the bottom. We're going to see this in lab 17 as well. So for instance, this third tube is a facultative anaerobe, and we have dense growth at the top and more sparse growth below. Notice that there's no growth at the very, very top, and that's because, again, gravity will cause the organisms to settle or kind of sink below over time. This, for example, is a microaerophile. It's not just below the top, but a little bit lower because of that sinking. When we're working with obligate anaerobes, we have to make sure the environment is free of oxygen, devoid of oxygen. So in our lab, we're going to use an anaerobic jar that will contain a packet containing a catalyst, such as palladium, that will get rid of oxygen and produce water. And usually we'll use also some kind of oxygen indicator to show when the environment becomes anaerobic. This is another look at an anaerobic box, but we don't have this in our lab, unfortunately, yet. All right, so for the oldest bacterial lineages, they should be anaerobic because early Earth had no oxygen in the atmosphere, so these first microbes were anaerobic. Aerobic metabolism only evolved later after photosynthesis occurred by organisms like cyanobacteria when they produced oxygen through photosynthesis. For the second one, which bacteria grow at the top, which bacteria grow at the bottom. So the top would be obligate aerobes that require oxygen for growth, and at the bottom they would be obligate anaerobes because there's no oxygen there, and remember, obligate anaerobes are killed or inhibited by oxygen. And the reason for this is because those obligate anaerobes, they don't have those enzymes like superoxide dismutase, catalase, or peroxidase to detoxify those reactive oxygen species. So examples of lab tests used to determine aerotolerance or aerotolerance-related reactions include the thioglycolate tubes we saw, the anaerobic jar we just saw, and we can also use candle jars for capnophiles. Here, this is what we're going to see in lab as well. We put our plates inside a glass jar, and I'll put a candle at the top and just simply close the jar. That'll lower the oxygen levels and raise the CO2 levels, perfect for capnophiles. And the next slide, I'll show you the catalase test. This will detect the presence of catalase enzyme and helps us differentiate between staphylococci and streptococci. So the catalase test helps us differentiate organisms by the enzyme catalase, and catalase catalyzes this reaction. It takes hydrogen peroxide and converts it or neutralizes it into water and oxygen gas. And we can see a positive result if we see oxygen gas bubbles. So here, there are two smears of bacteria on the left and right of this slide, and they add a few drops of hydrogen peroxide. If there's no bubbling, it's catalase negative. If there is bubbling due to the formation of oxygen, then it's catalase positive. So a lot of my students will say, why couldn't I have done the catalase test when I was performing my morphological unknown? That's because this is a metabolic test. We're looking for the presence or absence of this enzyme, whereas for our morphological unknown, we're looking at physical characteristics like shape, or if it was acid-fast, etc. So streptococci happen to be aerotolerant anaerobes and catalase negative, whereas staphylococci, these are facultative anaerobes and they're catalase positive. And our book gives us an interesting clinical example of a bus driver who had developed type 2 diabetes over 10 years ago, got a scratch on his foot that didn't heal and became increasingly worse. So his daughter noticed a discoloration and oozing of pus, and he finally went to the doctor and was rushed to the operating room. So the concern was gas gangrene caused by Clostridium perfringens over here. And what you worry about is that, remember, these are obligate anaerobes, but in dead tissue will no longer be supplied with oxygen by blood vessels. So they provide anaerobic environments that are perfect for Clostridium to grow and release endospores and for those endospores to germinate. Okay, so what do we do to detect catalase? We add hydrogen peroxide to the sample. So H2O2, because if catalase is present, it's going to break down hydrogen peroxide into water and oxygen, producing visible bubbles, those gas bubbles of oxygen. For the second one, the candle jar, the candle flame will burn most of the oxygen and increase carbon dioxide levels, and that creates a microaerophilic and capnophilic environment, which means low O2 and high CO2 that supports the growth of bacteria like Neisseria. All right, let's recap 9.2. How do we interpret visual data demonstrating oxygen and CO2 requirements? So we can use a thioglycolate tube or something similar where oxygen is highest at the top and lowest or no oxygen at the bottom, because microbes are going to grow best where their optimum CO2 or O2 level occurs. So in these thioglycolate tubes, we're looking at oxygen. Only growth at the top is a high oxygen requirement. If they grow throughout, but denser at the top, that means they can use oxygen but can also grow anaerobically. If they grow only at the bottom, oxygen is toxic to these bacteria. If they grow just below the top, you know that they need lower than atmospheric levels of oxygen. If they grow in a candle jar, you know they are capnophiles and require higher levels of CO2. For the next one, identifying and describing different categories. So we talked about obligate aerobes. They have to have oxygen, so they only grow at the top. They use aerobic respiration, and they lack the anaerobic enzymes. For obligate anaerobes, oxygen is toxic, so they only grow at the bottom. They don't have enzymes to neutralize reactive oxygen species. For facultative anaerobes, they prefer oxygen, but can still grow without it. So they grow throughout, but denser at the top, and they can switch between aerobic respiration and fermentation. For aerotolerant anaerobes, they don't use oxygen. They tolerate it though. So they grow evenly throughout, but usually sparsely. And they rely on fermentation, and they usually have one or more reactive oxygen species neutralizing enzyme. And for microaerophiles, they need low oxygen concentrations, lower than atmospheric levels, between 1 to 10%, and they usually show a thin band of growth just below the surface. High oxygen would be inhibitory to these. Capnophiles prefer elevated CO2 concentrations, and often that's in combination with lower than atmospheric O2 concentrations. So they grow better in a candle jar, or we can even use something like a CO2 incubator. High CO2 stimulates metabolism in these microbes. And then for the third bullet point, examples of microbes for each growth category. Obligate aerobes include like Mycobacterium tuberculosis. Bacillus subtilis is also an obligate aerobe. For obligate anaerobes, we talked about Clostridium, and even the Bacteroides bacteria in our gut are actually also obligate anaerobes. For facultative anaerobes, these are things like E. coli or Staphylococcus aureus. For aerotolerant, we talked about Lactobacillus. So these are found in fermented foods or even in the vagina. For microaerophiles, we have Helicobacter pylori that causes ulcers. And for capnophiles, we have Neisseria, such as Neisseria gonorrhoeae that causes gonorrhea. Now let's switch to 9.3 and look at the effects of pH on microbial growth. You may have noticed that acidic foods last longer than others, and that's why they've been a part of our human diet for centuries. They're harder to spoil. The reason for that is because most microbes that spoil food are neutrophiles. They grow best at a neutral pH, so they do not tolerate the acidity found in our acidic foods. Let's review pH really quickly. pH 7 is neutral, anything above that is basic, and below that is acidic. Extreme pH is bad because it can disrupt DNA, lipids, and especially proteins, leading to denaturation of proteins. Each microbe has a minimum, optimum, and maximum pH for growth. So for instance, for neutrophiles, you can see this end is the minimum pH, and this one's the maximum pH. Over here, we would have the optimum pH for max growth. Most bacteria are neutrophiles and prefer or grow best at a neutral pH. These include E. coli, Staphylococci, and Salmonella. Some bacteria are acidophiles. They grow best at a low pH, such as around pH 3, such as the Archaean cephalobus, or Lactobacillus that we find in acidic foods or even in the vagina. For alkalophiles, they grow best at a basic pH, and these include Vibrio cholerae or another Archaean like Nitronobacterium. Remember, many archaea actually can survive in more extreme environments, such as acidic environments or basic environments. And the reason the acidophiles or alkalophiles can survive more extreme environments is because they have adapted to these environments over time. So acidophiles, many of them can pump out the extra acid, these hydrogen ions, and some of them have altered their protein and membrane structure to survive in these high H+ or high acidic environments. For alkalophiles, many of them use sodium gradients instead of proton motive force. Most human pathogens are neutrophils. They prefer neutral pH levels. But Helicobacter pylori, which is also a neutrophile, can survive stomach acid. And they do so by producing an enzyme called urease. Urease catalyzes this reaction, converting urea into ammonia and carbon dioxide. Ammonia can bind to the acid in the stomach, forming ammonium. So this will raise the pH of the environment. We call that the microenvironment. So it's the environment right around or surrounding the bacterium, and that allows them to survive and potentially cause ulcers and gastric cancer. So extreme pHs can change the ionization of amino acid groups in our proteins. Remember, amino acids are the monomers of proteins, and they can disrupt hydrogen bonding. We saw that in secondary, tertiary, and quaternary structure. So this can impact protein folding, leading to denaturation and loss of enzymatic activity if that protein was an enzyme, or structural integrity if that protein was part of the cell structure. For the second one, pH adaptive type of bacteria. So human pathogens are generally neutrophils, right? Most of our body is at a neutral pH. So these bacteria, these neutrophils, grow best at a near neutral pH, around 7, like our human tissues and our blood. All right, let's recap 9.3. For the first one, pH requirements. Each microbe has a pH range it can tolerate. The minimum pH is the lowest pH at which it can grow. Below this, growth stops and the cell's damaged. Optimum pH was at peak, the pH at which growth is fastest. And maximum pH is the highest pH at which it can still grow. Above this, growth stops and proteins and enzymes might denature. So these three values together define that pH growth curve for an organism. For the second one, the different categories, we said acidophiles grow best at low pH, less than pH 5.5. So you might find these in acidic hot springs or acidic foods. For neutrophils, they grow best at neutral pH, around 7, or the range is usually about 6.5 to 7.5. So this can be in our human tissues and neutral environments. Alkalophiles grow best at high pH, around 8 through 10.5. So you'll see these in alkaline lakes or alkaline soil. For the third one, examples of microbes for each pH requirement, we talked about for acidophiles, lactobacillus, or cephalobus, that archaea. For neutrophils, E. coli, staphylococci, salmonella. And for alkalophiles, Vibrio cholerae, as well as the archaean Natronobacterium. That takes us to the end of part 2. In part 3, we'll continue with temperature and other environmental conditions that affect microbial growth.